All right, so before we get started, I just wanted to make sure to remind everybody of our midterm that is coming up. It's coming up on Monday, October 31st, 6 to 8 p.m. And this will be held at the Academy, okay? And it's worth 20% of your final mark. And also last time, one of the students was asking about when is the project due. So I think I'm gonna hold the student presentations on Wednesday, November the 16th. So this is right after we've finished our 4th unit, our unit on homeostasis. And before we get into population dynamics, we'll do the student presentation, okay? Oh, I guess I might as well also let you know that then your final exam date is also Wednesday, December 7th. And this will allow us a little bit of time to, between the last lesson, which is on Monday, November the 28th, to have some time to study, to hold a tutorial session or two if necessary, and then have the exam on Wednesday, December the 7th. Okay, I hope everybody takes note of those days and clears your schedule. And again, the final exam is also in person, held at the Academy. And I will be, excuse me, I will be there. I will be there as well. All right, now that we've taken care of a little bit of that, also, yeah, to remind you that we're a couple of lessons away from the end of Unit 3. Unit 3 assignment is due on October 30th. And that we will have a tutorial session on October 26th prior to our exam, which will be our midterm exam, which will be on Monday 1st. So we will hold a tutorial session. And but even before then, if you have any questions, if there's things that you'd like to review or talk about, I am available. Just reach out to me. All right, so lesson 11. So this lesson is on biotechnology and gene modification. And as you recall from our previous lessons, we talked a little bit about, excuse me, about the DNA molecule, about a bit about its structure. We certainly talked about how that DNA information is then transcribed and translated into a working protein. But really, the scientific knowledge in the field of molecular genetics has really advanced rapidly over the last few decades, and especially it has really accelerated even in the last 5 years. The knowledge base of this has certainly grown so much. So what we are going to talk a little bit about today is just to introduce you to some of the tools that scientists have at their disposal in order to manipulate DNA and as a result manipulate the proteins that are encoded by this DNA. These tools are often living organisms and also biological molecules, and scientists have figured out how to make use of the natural properties of these to bring about the kind of changes that are of interest to them. What we'll talk about today specifically is we're going to talk about the role of some of the cell components that are used in biotechnology, and such as the role of plasmids and restriction enzymes and recombinant DNA. We're going to look at some examples of gene modification, look at the process of DNA sequencing, and also the polymerase chain reaction. And these are just introductory tools that are available and are really the most commonly used sort of day-to-day tools in a molecular biology research laboratory. And what you would like to keep in mind as we're going through today's lesson is to think about how can living organisms be used to help scientists manipulate DNA? How can cell components be used as biotechnology? And what is the PCR, the polymerase chain reaction, and how is it used? Right, okay. So we're going to talk a little. One of the primary tools that are available to us. And when it comes time to manipulating DNA and changing it, one of the most basic and most important ways to be able to do that is to be able to take out a piece of DNA that we're interested in out of the rest of that greater genome, and to take that piece and to be able to manipulate that. Now, keep in mind that human DNA that we have in our cells is extremely large. It's approximately 3 billion base pairs worth of information. And so really the entire genome or the entire set of genetic material, 3 billion base pairs, would be very, very awkward to work with if you're only interested in studying and looking at a small segment or one gene or even a small set of genes. So restriction enzymes, or the other name for them is endonucleases, they are very useful because they can act as somewhat of a molecular scissor to help us cut that double-stranded DNA at very specific points. So these enzymes, these restriction enzymes, naturally occur in bacteria, and each one of the different bacterial species has got a specific set of them. And these enzymes in bacteria work as a sort of a very primitive immune system, because just as there are viruses that attack human cells, there's viruses that infect bacterial cells. And so when the bacteria detects the presence of these viral material, this viral DNA, it produces the restriction enzymes that then work by cutting up this foreign DNA and therefore ending that infection. So each one of these enzymes, and I will bring you down to this chart here that has some examples of these enzymes. So each one of these enzymes that we have here, here's some examples of different enzymes, and they all come from different organisms. For example, the enzyme EcoR1 comes from E. coli. Small one comes from Sriracha merskens. I'm not sure how to pronounce that one, sorry. And then there's also Ali1 from Arthrobacter luteus, and Sal1s from Saccharomyces albus. Hindi 3 is from Haemophilus parainfluenza. And so each one of these has a specific recognition site. So each one of these has a specific recognition site, and if the viral DNA that has entered the bacterium then carries any one of these recognition sequences, and these are the specific recognition sequences that these enzymes will recognize and be able to bind to, the enzyme will attach itself at this site and then cut that DNA. And these red arrows point to the specific site that is cut by these enzymes once they recognize and attach themselves to these DNA sequences. So it cuts this DNA into pieces, and because obviously in each type of bacteria, there's more than one type of restriction enzyme, so it really works well in defending itself against viral infections. And so it has all of these different, for example, E. coli might have many different restriction enzymes, EcoR being one of them that we're showing here as an example. But if you can see, each one of them has a specific recognition site. So if you can imagine, this viral DNA is going to have numerous recognition sites scattered across it, and it's just going to get chopped up into pieces by these restriction enzymes. So now scientists have actually isolated these restriction enzymes, many different restriction enzymes. And as a scientist working in the lab, there's catalogs of these restriction enzymes, and they're available at your disposal as a tool to use. And you can, you have certainly a huge range of these available to you in your laboratory's freezer. And if there's any specific ones that you want, you could buy them from companies that sell these things. So you can really treat these as tools. So these enzymes can be used by scientists under controlled laboratory conditions to target specific sequences and to cut up DNA into fragments, smaller fragments, that then become more manageable and easier to study. And if you look at the name of each one of these, what you'll notice is that the enzyme itself is partially named after the bacteria from which it originates. For example, the Hindi 3 is named after the Haemophilus parainfluenza. The H part represents the genus Haemophilus. The IN part of HIN represents the influenza part in that name. And the D part represents a strain, a specific strain, the RD strain. And the number 3 means that it was the third enzyme isolated from this strain. So there's actually Hindi 1, Hindi 2, Hindi 3, and each one of them has got different recognition site that gets cut by their specific enzymes. And there's 2,500, probably actually now more, I'm sure more. This number might be somewhat outdated now, that are available. And there's about 200 different cut sites, probably more. And this gives scientists a lot of choice as to what type of molecular scissor can they use depending what sequence they want to cut. So another technique that is very useful in the molecular biology lab is a technique of gel electrophoresis. And once this DNA has been cut up into smaller fragments, actually maybe before I get into gel electrophoresis, there's a little video maybe we can watch together very quickly. Yeah, why don't we watch this one quickly and then we'll talk about gel electrophoresis. So let's just let me just bring over a set of videos that I have lined up. Okay, so this one describes how, I just wanted to show this to you, just so that you have an idea of restriction enzymes and how they wrap themselves around the DNA, and how by actually once they recognize and bind to the DNA, their conformation changes, and they make the right bonds and disrupt certain bonds on the DNA in order to carry out the cleavage activity. So let's watch this together.
This is a structure of a restriction enzyme called MBE. What you'll notice is that this has a shape somewhat like a hand. It's sort of, you could imagine this to be a thumb. These could be fingers, and it has this shape because this is going to grasp a DNA molecule.
So this is a protein, right, guys? This is an enzyme. This is a protein. And when you look at it, you can recognize from our lecture on the structure of protein that there is some helical structures here, and there is some beta sheets. These are secondary structures that you find in proteins. And all together, they form this, give this conformation, this sort of an overall shape to this restriction enzyme. And then what they're going to show in this picture is in the pink is this structure of the enzyme when it's not bound to DNA. And then they're going to show you sort of a piece of DNA. They're going to model a piece of DNA, and they're going to show how the conformation changes once it's bound to the DNA. And you're going to see that new conformation shown in orange. I know the volume on this video is not too good, so I'll try and pause and fill in as much as I can.
It's going to grasp the molecule. It's going to, once it's grasped it, it's going to slide along it until it encounters its recognition sequence. Pink is without the DNA, so this is the free enzyme on its own. As it would exist in the cell when it was about to encounter the DNA molecule but hadn't. Now in orange, we're looking at the structure of precisely the same protein when it has encountered DNA. The structure changes when it attaches to DNA. Notice this loop here, this loop here. Both of them close.
So here now, in order to show what he's talking about, notice how this loop here and this loop here have closed once the enzyme is bound to DNA. He's overlapped these structures. And these are things you can certainly carry out. And these are modeling programs for modeling proteins based on structural data. And you can overlap these structures and really, as a scientist, appreciate the conformational changes that take place when this enzyme, this restriction enzyme recognizes and binds to its specific recognition sequence.
So in orange now, we have the structure of the same protein, but now this is found in DNA. And there's tremendous movements of regard. This enzyme has wrapped itself almost completely around the DNA. This is the DNA molecule that the enzyme is with. This has the sequence CCNOT, which is the recognition sequence of this enzyme. Now I put in the enzyme, and you'll notice that the enzyme is intimately associated with the major group of the DNA. This is the surface structure now of the enzyme when it's bound to DNA. The protein has almost completely encircled the DNA. There's just a small group which is following the contour of the double helix. The protein follows the helical terms, right? And almost completely encloses the DNA.
Okay. So I just wanted to make sure we had an idea about that. And we can go back to our so that was one example of these endonucleases. So then the technique of gel electrophoresis is another important one to talk about. And so once you've used these restriction endonucleases to cut up your DNA into smaller fragments, you need to have a way of actually separating the piece that you're interested in out of all the other pieces, and to separate it out of all the other ones to, and you can do this using gel electrophoresis. And the way to do this is to take advantage of some of the chemical and physical properties of DNA, keeping in mind that DNA molecule has an overall negative charge over it. And this is because of that sugar phosphate backbone, those phosphate groups that line the DNA backbone carry an overall negative charge, giving that DNA molecule an overall negative charge. And the other thing to take advantage of is the size of each one of these DNA fragments. You can imagine you're going to have DNA fragments of various sizes. And so the size is obviously consistent with how many base pairs are in each one of these pieces. So you can use gel electrophoresis as a way of separating these fragments according to their size and using the fact that they're negatively charged as a way of moving them through the gel. So the gel itself, I can describe as, here, I'll go to the picture. Maybe now this would be better to look at here. So the gel itself is this slab, this rectangular slab of a gel, and it's made out of a polysaccharide called agarose. And agarose is extracted from kelp, seaweed. And so when you mix this agarose with a solution of electrolytes and melt it, and then let it cool and set, it solidifies into this sort of a gel material. And this gel is quite dense. If you can imagine, it's kind of like Jell-O, but about 5 times more dense. So it's kind of like a very firm kind of a gelatin kind of a consistency. And if you think about this gel at the molecular level, it's composed of a series of molecules that are cross-linked together to create kind of a mesh with tiny pores. And so the DNA that's been cut, when it's physically actually placed in the gel, and this is what you're seeing here, somebody's pipetting some of this DNA material into these wells that are in the gel. And once you can connect electricity to the 2 ends of the gel, so that you, in this contraption, you will have a negative electrode at this end and the positive electrode at this end of the molecule. So the current will run, making the negatively charged DNA want to migrate towards the positive electrode. So as this as you turn on the electricity, when the electrical current is applied here, the DNA fragments will move from the negative electrodes toward the positive electrode. And as they go through and they're weaving their way through this gel, through this porous gel, the smaller fragments will be able to move through those pores faster. And the larger fragments will get hindered, will get held behind somewhat and run slower. So in this way, you will get this separation of the DNA fragments based on their size, with the smaller fragments moving faster, so moving further ahead, and then the larger fragments that, given the same time, will have moved less. So if you want to maybe think of this as an obstacle course and think how quickly one person can make their way through an obstacle course as compared to if you have a group of people all holding hands together, they're going to have a harder time and they're going to be slower moving through this obstacle course. A smaller person or maybe 1 or 2 people are going to have a faster time going through the obstacle course than a whole group of them. So once this DNA has separated, then what happens is that this gel actually gets exposed to a dye that will allow the DNA fragments to become visible. And the most common dye that's used is ethidium bromide, which is a molecule that actually attaches itself to the rungs of the DNA ladder. And when it gets exposed to the UV light, this ethidium bromide will fluoresce, will glow, and it will light up the fragments for you. Now, nowadays, they've developed other options other than ethidium bromide, because if you can imagine a chemical that is able to attach itself to the DNA is quite dangerous to work with. And certainly in the days that we were working with ethidium bromide, you had to be very, very careful handling bromide. It's a mutagen because it attaches itself to DNA. Now there's safer alternatives that allow you to visualize DNA. So I've got a video lined up, which will really show all the concepts that I talked about in an animated fashion that will help you see gel electrophoresis a lot better. Okay, so here we go.
One of the most common methods for visualizing and identifying DNA fragments is called agarose gel electrophoresis. With just a small gel like this, which feels a bit like thick jello, we can explore the world of DNA. I'm Dr. Alex Danis with Mini PCR Bio, and today we're going to explore this important laboratory technique. Electrophoresis is a long word that means carried by electricity. In the lab, it refers to the movement of molecules like DNA, RNA, or protein mobilized by an electric field through some kind of substance. One of the most common uses of electrophoresis is to use an agarose gel to separate pieces of DNA in order to visualize them and determine their sizes. Today, we're going to take an in-depth look at that process. In any gel electrophoresis setup, there are two electrodes, a positive electrode near one side of the gel and a negative electrode near the other. When we turn the power supply on, the two electrodes create a difference in charge on the two sides of the gel. This creates an electric field through the gel. How electrophoresis separates molecules is affected by the molecule's size and charge. Charge causes a molecule to move through an electrophoresis gel as it is attracted to the oppositely charged pole. Positively charged molecules are pulled towards the negative electrode, negatively charged molecules are pulled towards the positive electrode, and neutral molecules won't move at all. Because DNA is negatively charged, it will always move towards the positive electrode. In an electric field with no obstacles, all fragments of DNA would move at the same speed towards the positive electrode. But scientists want to separate DNA fragments by size. So in gel electrophoresis, they add them to a gel full of irregular holes and pores, almost like a sponge. When moving through such a complex substance, the size of the DNA fragment determines how quickly it moves. Short pieces of DNA will move quickly through the gel, zipping through all of the pores without difficulty, while long pieces of DNA will encounter more hurdles moving through the small pores, making them move more slowly through the gel. The short pieces will travel quickly towards the far side of the gel, while the longer pieces will move more slowly and travel a smaller distance in the gel. In this way, we can use gel electrophoresis to separate a DNA sample based on size. Now that we know what happens in gel electrophoresis, let's talk about how we actually perform this technique in the lab. First, we need to make our gel. DNA electrophoresis gels are usually made from agarose, a long polysaccharide molecule extracted from seaweed. Making a gel is a bit like making jello. You dissolve a powder in a liquid, boil it, and allow it to cool and thicken in a mold. To create an agarose gel, we dissolve agarose in an electrophoresis buffer, boil it, and add it to a gel casting tray. Typically, agarose comes as a powder, but it also sometimes comes as a pre-weighed tablet. We usually also add a DNA stain at this step to make visualization of our samples easier later on. To learn more about gel pouring, you can watch our How to Pour a Gel video linked below. In order to see our sample in the gel, we first need somewhere to add the DNA. To accomplish this, we place a comb at one end of the gel. Here, we've poured two gels. When the gel cools and hardens, we pull this comb out of the gel and small wells are left behind. The wells are like little pockets in the gel, and they're how we put our DNA into the gel at the start of an electrophoresis run. When the gel is cooled, we place it into a gel electrophoresis chamber. We cover the gel with running buffer, which helps to conduct electricity. Before adding a DNA sample to our gel, we mix it with a loading dye, though often this dye is already added. This dye is more dense than the running buffer that the gel sits in, so when we carefully pipette the mixture into the wells at one end of the gel, the DNA will sink to the bottom. The color of the dye lets you see that your samples have been loaded properly into the wells. The dye is also negatively charged and tinted so that we can more easily track the movement of molecules through the gel. Once our samples are loaded, we will close the lid and turn on the power. Here, we're using the blue gel from Mini-PCR-Bio, which combines an electrophoresis chamber and visualization system. The blue gel will take about 20 minutes to run. Once the gel has run, we need a way to visualize our DNA. Many different DNA stains can be used for gel electrophoresis, but they all bind to DNA and make the DNA visible. These stains are different from the loading dyes we discussed before because they bind directly to the DNA. Using fluorescent stains, we can visualize our DNA just by turning on a safe blue light. Here in our blue gel electrophoresis system, we can simply turn on the light to visualize our DNA and directly capture images using a smartphone without transferring the gel to a second location. Using a dark hood can help to see the bands in a bright room. We can even use our phones to make a movie of our gel as it is running. Because the DNA runs in a straight line from the well towards the far end of the gel, we call each region of the gel that a single DNA sample runs through a lane. Here, the lanes in the middle of the gel represent the DNA in each of our samples. Here, we have a lane with two bright stripes called bands. Each band is actually billions of DNA fragments, all of the same length, that all moved through the gel together at the same speed because of their identical sizes. All of this DNA started in the well here and traveled in this direction through the gel. How quickly a DNA fragment moves through the gel compared to another can tell us how big they are in comparison to one another. For example, in this lane, this band, labeled A, has moved farther through the gel than band B, so I know that the DNA in band A is smaller than the DNA in band B. But usually, we want to know the actual length of the fragments of DNA we are separating. To better assess the size of our DNA fragments, we typically add a DNA ladder to one of the wells. In this gel, the ladder is in the leftmost lane. A ladder is a sample of prepared DNA fragments of known lengths that acts like a ruler we can later compare our DNA samples to. The smallest fragment, which is only 100 base pairs, will move the farthest through the gel, then the 200 base pair fragment, the 300 base pair fragment, etc.
Yeah, so here in this lane, this DNA ladder that you see is a standard mixture of pieces of DNA of certain standard sizes. So you can use this as a ruler almost for comparing your bands and trying to estimate how big these other pieces of your pieces of DNA that you're interested are compared to the standards that are here in this DNA ladder. So for example, you can see in this lane, this one is almost 700 base pairs, maybe slightly larger because it almost looks like it's running just slightly higher than that. You can see, for example, here, these three bands are the same size. They look like they're running at the same point in the gel. And if you were to estimate the size, they're bigger than 200 base pairs. They're smaller than 300 base pairs. But excuse me, you as a scientist have an idea of what size of a band you're looking for, and is this your 250 base pair band that you were interested in or not? And this one might be a 400 base pair band that you were interested. This one might be an 800 base pair band, for example.
To determine the size of each band, we can compare them to our ladder. One band contains DNA that is about 250 base pairs long, and the other contains DNA that is about 150 base pairs long. If we look in the next lane, we can see one band that is about 400 base pairs long. DNA electrophoresis allows you to check and see if a piece of DNA of a specific size is present in your sample or not. This can help you figure out the likely identity of that piece of DNA, helping to confirm an infection, identify an individual by DNA fingerprinting, and more. It is especially useful after PCR to confirm whether or not you amplified a desired piece of DNA. You can learn more about PCR by watching our video linked below. gel like
Yeah, and the other thing that you can do that is also useful is sometimes you can actually physically cut out this band from your gel and extract it from the agarose and purify it, and then you have purified that fragment of DNA. You can separate that piece of DNA. Okay. So then the other important molecular biology tool are plasmids. Plasmid is another piece of the cellular machinery that is very useful to scientists. They're small circular pieces of double-stranded DNA that are found in the cytoplasm of different types of bacteria. And this plasmid DNA is different from the bacterial genomic. They are separate from the bacterial genomic. They're small circular pieces of DNA. And their natural role is to provide some extra genetic information for the bacterial cells. And in many cases, they actually carry genes that allow the bacteria to have some antibiotic resistance. And now, because they are separate from the genomic DNA and much smaller, these plasmids are very easy to remove from the bacterial cells. And then you can use restriction enzymes to actually cut open these plasmids and to add various genes or various other pieces of DNA to it. And then when that plasmid is put back together and put back into that bacterial cell, the cell's machinery will take care of expressing whatever piece of foreign DNA or whatever piece of gene that you're interested that you put in there, and actually carry out and synthesize that protein for you using its own transcription and translation machinery. And this is really at the heart of recombinant DNA technology, where you've taken genes of two different species and put them together in one organism and caused it to express a foreign a gene in a particular organism. So I'm going to use, again, another video to demonstrate this idea for you.
Any newcomer who joins a molecular biology lab will undoubtedly be asked to design, construct, or modify a plasmid. But what exactly is a plasmid? And why is it so useful in the lab? At their most basic level, plasmids are small, circular pieces of DNA that replicate independently from the host chromosomal DNA. Compared to the millions or billions of bases that make up the entire genome, plasmids typically contain a couple thousand at most. They are relatively small, stable, and easy to manipulate. So where do plasmids come from? In nature, they're found in microbes like bacteria. In the 1940s, scientists began noticing that there are heritable cytoplasmic factors that could be transferred between cells, but there was no consensus on what they were or what to call them. It wasn't until 1952 that Nobel laureate Joshua Lederberg coined the term plasmid, a combination of the words cytoplasm and id, Latin for it. In nature, plasmids often contain genes that provide a competitive advantage, giving its host bacterium an ability that it didn't have before. These benefits include antibiotic resistance, the wherewithal to survive in harsh environments, and even the ability to wage war to gain an environmental advantage. But plasmids aren't just useful to bacteria. Because they're incredibly easy to manipulate, They've also made themselves indispensable to life scientists and bioengineers. Plasmids created in the lab are known as constructs or vectors. To understand what plasmids can do, let's break down its parts using a plasmid map. All plasmids contain an origin of replication, or ORI. It tells the plasmid where to begin replication. Plasmids often contain genes that are advantageous for survival. One of the most common, naturally occurring types of genes is antibiotic resistance. When used in the lab, antibiotic-resistant genes allow scientists to separate out cells that contain plasmids from those that don't. The wonderful thing about plasmids is that they can be easily engineered and can introduce foreign DNA into cells through electroporation or other methods. Many plasmids are designed so scientists can insert genes they want to be expressed in organisms. One way to do this is through sites. Restriction enzymes recognize these sites and cut out like molecular scissors. Then, a different gene can be inserted into the site. Often, these restriction sites are located in what's called a multiple cloning site, a short segment of DNA that contains several restriction sites. This adds flexibility to the cloning process. How is the inserted gene expressed? A promoter site, which is upstream of the inserted gene on the plasmid, acts as a green light that allows gene transcription. RNA polymerase binds to the promoter, moving along the strand. As it moves along the strand, it creates a new strand of mRNA expressing the gene. The plasmid cloning process is very versatile, so there's a whole array of genes that scientists can introduce into the cell. For example, if you wanted to track a specific species of bacteria in a population, you could insert a GFP-expressing gene into a plasmid and transform it into your bacteria. Your cells will fluoresce, making them easier to find. Or if you're trying to study the effect of a specific
Yeah, GFP stands for green fluorescent protein, and that's why they were saying that if you put in a GFP, it will fluoresce. So green fluorescent protein is GFP. Just in case you're wondering.
your cells will fluoresce, making them easier to find. Or if you're trying to study the effect of a specific protein on a phenotype, you could insert the gene of interest into the plasmid, move this into a cell, and look for changes in the cell. We hope you enjoyed this video in our Plasmids 101 series.
So let's talk a little bit more about gene cloning and how it's done in using plasmids. So one of the important techniques is gene cloning. So this involves using these bacterial plasmids and inserting a piece of foreign DNA from some other organism and using the cellular machinery of the bacteria to use it to express whatever protein that gene is encoding under controlled conditions. So in here, the gene that you are interested in is cut out from the genome of its original species using whatever endonucleus that's suitable for cutting that piece of DNA out. And then you can use gel electrophoresis to isolate this piece of DNA from the rest of the fragments. And then you have a plasmid, which you will remove from your bacteria, purify, and then cut that with the same endonucleus nuclease so that you can have these two sticky ends bind together. And so, so if you cut both of these with the same restriction endonuclease, with the same restriction enzyme, and then mix them together due to pairing of the base pairs and the recognition of the base pairs with each other, they will find each other and stick together. And then you add another enzyme called DNA ligase to, in a way, glue these fragments up to seal the backbone of the DNA. And then you have this recombinant piece of DNA. You have the plasmid with a gene fragment from a different organism inserted in it. And you can introduce this plasmid into a bacterial cell through transformation or electroporation. There's various techniques. And then off you go. That bacterium will then, as it divides and grows and creates multiple copies and replicates this DNA and expresses the protein that that piece of gene encodes. So that resulting protein can then be isolated from the bacterial culture and purified for further study. And one of the really best examples of how this technique has been used is in producing human insulin for diabetics. And in the past, diabetics used to have to inject themselves with insulin purified from pigs, so pig insulin. And so insulin is a molecule that is made in the pancreas. So the pancreas from these pigs were literally collected from slaughterhouses and ground up, and the insulin was isolated and purified from them and used as a treatment for diabetics. But today diabetics use human insulin that has been produced in bacterial cells through recombinant DNA technology. So that human insulin gene is isolated and recombined into plasmid DNA. And once it's put back into bacterial cells, the bacterial cells start producing that human insulin, which then can be isolated and purified and made available to diabetics. So we no longer need to use pig insulin. We can actually use human insulin, which has much better results. So another tool in the toolbox of molecular geneticists is the use of RFLPs. And RFLP stands for restriction, fragment, length, polymorphisms. And this technique of RFLPs also makes use of the technique of gel electrophoresis, which we talked about, and the fact that DNA fragments of different lengths will move to different positions in the gel based on their size and their fragments. That's something we talked about and looked at. But in this technique, the DNA sample that's in question is cut up with numerous restriction enzymes to make a series of fragments. And then these fragments are run out on the gel to separate them out. And because of just the amount of DNA that's used, the fragments actually show up as a bit of a smear, somewhat of a smear on the gel. So actually, let me bring up the image here. This picture shows the RFLP process. So yeah, you take the DNA that you've extracted from your sample, you cut it up with restriction enzymes. In this case, not just one restriction enzymes, but many. The act of cutting up DNA with restriction enzymes, another word for it is also to digest the DNA. So after you've done your digestion with many restriction enzymes, you run the fragments on the gel, but they will appear as a smear because there's a large number of them. And so then what you do to be able to see those fragment patterns and make a more permanent record of this fragment pattern, you have a nylon membrane that you place over the gel, and the DNA fragments in the gel will move from the gel onto the membrane when an electric field is applied. You soak this nylon membrane in the solution that contains a radioactive complementary probe. And so these short complementary stretches of the radioactive DNA that are exposed to the membrane, and because of the complementary base pairing, these radioactive probes will bind to the DNA fragments on the membrane. And when this membrane gets exposed to an X-ray film, the radioactivity will leave, will create a mark on the film once you develop that film. But so what is different in this technique compared to just basic gel electrophoresis is that although they are very similar in that you take your DNA, you digest it, you run it on a gel, but because this technique is used to look at regions of DNAs that are different from person to person, and that's really what we're using, that's the power of this technique, is to be able to identify different people, for example, or different species. That's where this technique is different. So there's in the DNA, there's regions of DNA. We know that code for genes that are actually important to organisms, but also a large amount of the genetic code is essentially junk DNA. That at least at this point, a lot of it is considered to be just extra DNA, and the purpose for it is really not known. It seems to serve no purpose. So the parts that do code, those coding regions of DNA, those coding regions, because they code for certain proteins, there's not as much variations in those sections of DNA from individuals within the same species. For example, my gene for the protein hemoglobin will look very much like your gene from hemoglobin, but parts of our DNA that are not so well, don't code for anything, the junk DNA, the non-coding regions of DNA that don't seem to really affect the organism, if changes occur in the DNA, those regions seem to have a lot more differences between individuals in the same species. So, so we don't find as much change in the DNA code in the areas that actually code for enzymes and proteins, because any change in the DNA code will change the order of amino acids that make up the protein, and then the protein would not function properly. So mutations don't really tend to accumulate in the regions that actually encode for some sort of a functional protein or enzyme. Because if those regions were to collect mutations, they would lead to non-functional proteins and possibly disease or death in the organisms. But in the non-coding regions of the DNA, in the junk parts of the DNA, those parts don't seem to affect the organism as much. So if there are changes in those parts of the DNA code, because these regions don't seem to produce any protein, so generation after generation, these types of changes seem to accumulate and essentially have no effect in the organism. So because we are sexually reproducing organisms, the combining of our maternal and paternal DNA in the offspring leads to these unique nucleotide sequences in these areas of non-coding DNA because of those accumulated changes. And that means that if one of these regions, these non-coding regions, gets cut up with restriction enzymes, so in an individual, the pattern of that fragment would be different compared to the pattern of fragments from that same region in a different individual. So DNA fingerprinting, for example, is a good example of how this technique can be used. Just as our fingertips have fingerprints that are unique to each one of us, our DNA fragment pattern is quite individual as well. So this means that the DNA pattern of those fragments, from those non-coding sections, can be used to match a sample of DNA directly to an individual. And that's really the essence of DNA fingerprinting. And so, but because we have a huge number of people on our planet, it can possibly conceivably be possible for more than one person to have to generate the same DNA fingerprint pattern if we cut the DNA with only one restriction enzyme just due to chance alone. So that's why it's very important when you're doing this type of analysis, when you're doing DNA fingerprint analysis, that many different junk DNA areas are tested and cut up with multiple different restriction enzyme. And so by doing this, you will lower the chance of two individuals producing the same pattern. And you will lower this, this chance to 1 in a trillion or so by doing that. So, so yeah, this table just summarizes the different steps in RFLP analysis. So again, you take the entire genome, you digest it with restriction enzymes, you take that digested DNA, you run it on an agarose gel like we did up here, using gel electrophoresis. And then you take the gel, you place it against a nylon membrane, you subject it to chemicals that actually denature and open up that DNA into single-stranded DNA. You apply current that transfers that single-stranded DNA to migrate from the gel and bind to the nylon membrane here. Then you take the nylon membrane and you place it in a solution that contains some sort of a radioactive complementary nucleotide probes that have been especially engineered in their sequence to bind to specific regions of the genome. And then these radioactive probes will bind and hybridize because of their complementary sequences to the single-stranded DNA that is on this nylon membrane. And then this nylon membrane with its hybridized radioactive probes is placed against an X-ray film, and the autoradiogram that is developed has a banding pattern that gets visualized here. So that's just RFLPs in short. And then for homework, this is kind of a cool little question for you guys to try and answer and study. Suppose you have a camper that is caught by authorities frying a fish. He's caught a fish that is believed to be out of season. So he shouldn't be fishing that fish and shouldn't be eating it. But luckily there's still a piece of the fish that hasn't been cooked yet. And so the authorities want to make sure that this piece of fish is definitely the type of fish that they're questioning. So they do some fingerprinting on this fish and using RFLP analysis. And they have this restriction enzyme pattern. And so the gel pattern that's shown shows a certain story. This is the restriction enzyme from the type of fish that is out of season. This is the restriction enzyme digest from the DNA of the camper's fish. So can you tell by looking at and comparing these two parts of the gel whether the investigation should continue, or is the camper good? Has the camper caught an out of season fish, or has the camper caught, in fact, a different type of fish? So then the other very famous molecular biology tool that we have at our disposal is the polymerase chain reaction, or PCR for short. And this is a technique that many of you have probably heard a lot about over the last couple of years, especially since it's such a powerful tool for doing COVID testing, that do a PCR test on it. And this technique really is used to amplify a small sample of DNA to make it many times more and amplify it using that original DNA to make lots of copies and to increase the amount that is present so that then it can be detected and measured. So the natural process of DNA replication, and in fact, all biological molecules that are involved, can be controlled, and you can force replication to occur over and over again until you have multiple copies of DNA that have been made. So let's look at, for example, this is a PCR DNA synthesis cycle that we can visualize over here. So if you think back to when we looked at DNA replication in our previous lessons, think about what were the components that we needed to replicate DNA. Can anybody think of some of the components that we needed when DNA replication was happening in the cell? What did we need? Well, we first of all certainly needed the DNA itself. We needed a DNA template. We needed some DNA to act as a template. And then the other important thing that we needed was to have some DNA polymerase. That's the enzyme that actually makes new. And then if you think back, we also needed primers in order for the polymerase to be able to attach itself and to begin making the new strands. And in order to make new strands, what do you need? You need to supply it with free nucleotides to be able to take those nucleotides and string it together and to form new strands of DNA. So in PCR, you take all of these components, a template, the polymerase, the primers, and the nucleotides, and you put them all together into a test tube. But you can't really copy DNA while it's in the double-stranded form. So, because we don't have, instead of using helicase and gyrase, and those single-stranded binding proteins, those were all factors that were also present in the cell during DNA replication. Instead of using those, heat is used in order to separate the double-stranded DNA to make single strands. So the temperature that is used is 95 degrees. At 95 degrees, the DNA double strands will denature and separate. And once you have that single-stranded forms, then those primers which you have, which are complementary to the DNA sequence from the ends of the region that you want to copy. So for example, let's say this is the double-stranded piece of DNA that you want to amplify and make many, many more copies of. So you need to design primers for each end of that of the two strands. You need to design a forward primer and you need to design a reverse primer. So you need to design two primers that are complementary to both ends of each one of the DNAs, both strands. So once you've separated these two strands of DNA, you need to make sure that the primers will bind through complementary base pairing. And there we go. That's better. So the primers will bind because their base pairings are complementary. You've designed these in a way to bind and recognize this stretch of DNA here, and to bind and recognize this stretch of DNA here. Then the polymerase will come and attach itself to these primers. And we'll use these strands as a template. And given that also in this solution, you have free nucleotides, so you have A's and T's and C's and G's, it will string together all of these nucleotides to create the complementary, the new DNA in both of these cases. So DNA polymerase will start building the new strands of protein. So now the DNA is replicated, and you have double-stranded DNA again. So again, you need to heat these up in order to separate these and separate these double-stranded DNAs to make single-stranded DNAs, where the primers are going to recognize and bind to the corresponding ends. And again, DNA polymerase will bind and will copy. And so you'll make copies, the new DNA. And so in this way, you can repeat this cycle over and over and over again until your original DNA sample has been amplified again many, many, many times, to the point where eventually, after just doing, for example, 20 cycles, you might go from having a single copy of the DNA. After 20 cycles, you would have amplified it over 2 million times or so. So this technique seems pretty much straightforward if you think about it, except if you think about the idea of you are heating this DNA sample to 95 degrees Celsius. And if you think back to that discussion we had about enzymes having optimal temperatures and denaturing at temperatures beyond that, so at 95 degrees, most proteins, and specifically enzymes, will get denatured and lose their shape when they're exposed to high heat. So they won't be able to function anymore. So it means that at 95 degrees, polymerase would stop working after the first cycle, where it got the DNA and the polymerase and everything in the test tube got heated up to 95 degrees. So the solution, very good solution to this, has been to use a very heat stable or thermostable type of polymerase. And this polymerase is isolated from the bacterial Thermus aquaticus, which lives in hot springs. And they have a polymerase that is capable of tolerating and surviving and working and functioning under high heat conditions. And this polymerase is called Taq polymerase. So Taq polymerase is very important in PCR. And it's used for its ability to stay functional despite getting exposed to these high temperatures that melt and separate double-stranded DNA into single-stranded DNA. So the type of polymerase that is used for PCR is this thermostable Taq polymerase. Taq from Thermus aquaticus. So yeah, I have a video for you to watch for the PCR, the polymerase chain reaction. So let's go to that.
We live in a moment where genetics is helping us to understand more and more of the world around us, from untangling evolutionary histories to more precisely diagnosing and treating disease. But to do all of this, we need to be able to find and examine specific pieces of DNA. One of the fundamental tools that we use to do this is called PCR, or polymerase chain reaction. I'm Dr. Alex Danis with Mini PCR Bio, and today we're going to explore this important laboratory technique. PCR begins with a DNA sample, and DNA samples can be complex. Imagine the human genome with 3.2 billion base pairs. Trying to identify just one genetic sequence, which may be only a few hundred bases long, out of the entire genome of billions of bases, can be like looking for a needle in a haystack. PCR is a tool that finds that needle and then makes copies of it over and over until you are left with a pile of needles so large it's hard to find the original haystack. So how do we find our genetic needles in our haystack of DNA? A lot of it depends on the fact that our DNA is composed of two complementary strands that form a double helix. An A on one strand will always bind to a T on the other, and a C on one strand will always bind to a G on the other. This means that if you know the sequence of one strand, you always know how to replicate the other half. PCR is composed of three main steps: denaturation, which separates the two complementary strands of DNA; annealing, which specifies the region of DNA to be copied; and extension, which copies the DNA. These three steps will then be repeated over and over again. Now let's take a deeper look into how all of this works. The first step, called denaturation, involves separating the two strands of a starting DNA sample apart. This starting DNA sample is referred to as our template DNA. To do this, we heat the DNA up to about 95 degrees Celsius, almost reaching the boiling point of water. In just a few seconds, the high temperature causes hydrogen bonds between the two strands of DNA to break. After an initial longer denaturation to start off the PCR, each cycle requires a shorter denaturation, which can be as little as 10 seconds, but is often between 30 seconds and 1 minute. In the second step of PCR, we specify the region of DNA to be amplified. We do this using short pieces of DNA called primers. Each reaction will have two types of primers, forward and reverse. The forward primer will match a sequence on one strand of DNA at the beginning of the region we want to amplify. The reverse primer will match the end of the region on the other strand. Primers are typically around 20 bases long. When we cool our reaction down, the primers will anneal or stick to their complementary regions on the template DNA. Lowering the temperature allows hydrogen bonds to form between the primers and the template at areas where the sequence is complementary. We add lots of extra primers to increase the chances that they stick to the DNA rather than the DNA strands annealing back to each other. Annealing temperatures are typically between 50 to 65 degrees Celsius, but the exact temperature you use in your reaction depends on the specific length and sequence of the primers themselves. Our reaction may be allowed to anneal for as short as 5 seconds or as long as 30 seconds. Next, we heat the reaction back up again to extend or copy these new double-stranded areas where primer and template DNA have annealed. For this, we use an enzyme called a DNA polymerase, the P of PCR. The polymerase will find the ends of the short double-stranded regions of DNA where the primers have bound. It then moves along the DNA, base by base, in the 5' to 3' direction and adds the correct complementary DNA nucleotides as it goes. If it finds a C on the template strand, it adds a G. If it finds an A, it adds a T, and so on, creating a complementary strand of DNA. Our extension step can vary in time. When our target is long, we can extend this step to a few minutes long to give the polymerase time to copy the entire region. If we are copying very short regions, it may only need a few seconds. Extension typically happens around 72 degrees Celsius, the optimal working temperature of the polymerase. Of course, 72 degrees Celsius is hot, Our own body temperature is around 37 degrees Celsius. So if we used a typical human or bacterial polymerase, it would likely denature and stop working at these high temperatures. So instead, we use a special polymerase called Taq polymerase. This enzyme was originally found in thermophilic bacteria living in the hot springs of Yellowstone National Park and remains stable in our reaction even through multiple rounds of heating and cooling. All of these steps happen in a machine called a thermal cycler. Here, we're using a mini PCR machine from Mini PCR Bio. We place our DNA along with all the ingredients needed for the reaction into tubes and then place them inside. The thermal cycler will heat it up and cool it down just as we tell it to. After one cycle of these three steps, denature, anneal, and extend, we have turned our original one piece of target DNA into two. But we don't want to stop there. We repeat this cycle over and over again, now turning our 2 into 4, then 4 into 8, 8 into 16, and so on. The process creates new identical copies of DNA. We are exponentially amplifying our DNA. This chain of copying explains why it's called polymerase chain reaction. Most PCRs will run for about 30 cycles, which should give us about 1 billion copies of our original target sequence for every one copy we started with. For all of this copying to work, we need the exact right ingredients. So let's take a closer look at exactly what we put into each tube. First, we need our DNA template. Remember that this template is often a part of a complex DNA sample. Depending on the experiment, this DNA could come from almost anywhere, including a human blood sample, a water sample, a food sample, or even a bacterial swab. We also need our DNA primers, which specify which area of DNA we want to amplify. The DNA sample and the primers are unique to each experiment, but the rest of the reagents are the same in almost every PCR. We need the Taq polymerase, which will do the copying. We also need lots of spare nucleotides or dNTPs, the building blocks of DNA. These are the molecules the polymerase will add to the growing DNA strand. Finally, all this happens in a special buffer, which contains salts and ions that help keep the polymerase functioning. What we do with our new DNA copies after our PCR finishes depends on our experiment. We may insert it into a plasmid so that a bacteria will produce a certain protein that we are interested in. We may use it to do DNA sequencing so we can learn the exact sequence of nucleotides in our DNA sample. Often, we will run it on an electrophoresis gel so that we can visualize the size of our DNA fragment or confirm that the PCR worked as we expected. This ability to make billions of copies of a specific DNA sequence has made PCR a staple of the molecular biology lab for decades and allows everyone from students to scientists to investigate DNA in the world around them. Mini PCR thermal cyclers continue to make PCR accessible to users around the globe, from classrooms to remote field outposts, and even up in space. To learn more about mini PCR thermal cyclers and learning labs that you can use to implement PCR and molecular biology techniques in your classroom.
Right. So that was the overview of the polymerase chain reaction. Now, one more molecular biology tool is DNA sequencing. So DNA sequencing, or Sanger sequencing, this technique was developed by a scientist called Frederick Sanger. That's why it's called Sanger sequencing, as a way of determining the sequence of your DNA fragments. And what this really means is giving you the ability to actually read out the order of the nucleotides that code for that genetic information. And this technique of DNA sequencing also makes use of DNA replication components. So let's go ahead and look at those components. So when you're setting up DNA sequencing, what you have to do is you set up 4 test tubes, And in these 4 test tubes, you will have, in all 4, you will have your single-stranded DNA template. You will have a short primer. You would have your DNA polymerase. And then you will have the 4 deoxynucleotide triphosphates. You will have the DATP, DTTP, DGTP, and DCTP. So the A, T, G, and the C collectively all together, you call them the deoxynucleosides or the DNTPs. The N means basically all of these, the A, the T, the G, and the C. So in addition to these 4 components that you have in all 4 of your test tubes, each one of the tubes, you will put a very low concentration, and this is actually important that be low, a very low concentration of a different radioactively labeled, or nowadays fluorescently labeled, dideoxy analog of one of these DNTPs. And you have this in very low concentration. And I'll explain in a moment why this is, that it's important that it be present in low concentrations. So now you have all 4 test tubes that have everything that is required for DNA replication, plus one of these dideoxy analogs. For example, tube 1 will contain all 4 DNTPs plus dideoxyadenine. And your second tube will have everything plus dideoxythymine. Your third tube will have everything plus a low concentration of dideoxyguanine. And the fourth one will have the C, will have everything plus a small concentration of dideoxycytosine or DDCTP. And what's important about these analogs that you're putting in, each only having one type of, each one of these tubes only having one type of these analogs, is that these analogs are different in that their sugar is missing that hydroxyl groups on the 3rd carbon. And the 3rd carbon is where that phosphodiester bond would normally form as you are extending your chain during replication. So this means that if during replication, if one of these analogs is grabbed by polymerase, so instead of a regular DATP, it grabs a DDTP, which you don't have too much of in your tube. You only have it in very low concentration, so that if it happens to grab one of these and adds it in, what this will do is that this will immediately terminate the replication and end that chain, and it will stop it from growing anymore and stop the chain from forming, because you cannot add any more nucleotides after this. You can't bond any more nucleotides to the end of it because you are missing that hydroxyl group on the 3' carbon where that phosphodiester would form. And because these analogs we said are present only in low concentrations, then that means that only very infrequently and only by chance they will get added into that growing DNA strand, and only once in a while. So during the replication process, that DNA polymerase will use that single-stranded DNA and use it as a template to produce the complementary strand. But in each test tube, as those bases are required, they will only occasionally incorporate one of those dideoxy analogs. And so this means that in every test tube, you will be able to replicate the entire strand sometimes, and in other cases, only fragments of the whole strand will be produced. So as an example, if in your sequencing, you have this sequence, for example, and you want to sequence this, so you're going to be replicating this, and it's part of a mixture that contains all the all the regular requirements. So it contains the single-stranded DNA template. It contains a short primer that will recognize the sequence at one end. It contains the DNA polymerase, which will bind to the primer and carry out the copying, the replication. And you will have DATP, DTTP, DGTP, and DCTP. But this one, for example, it will be your A tube. So in this one, you will also add a very low concentration of DATP. So this is that analog that's missing the OH from the 3' carbon. So here, in this case, if you look at the sequence, based on this template sequence, you will be able to produce 4 possible complementary strands, because this template only has 4 possible T's in it, right? So if DNA polymerase comes and starts and binds and starts to copy, it will start copying using T, and then at this T, it'll add an A. And if it adds a regular A, it will be able to keep on going. But if it happens to grab one of these dideoxy ATPs, then it stops. It cannot keep extending that chain. So you will end up with a fragment that has stopped about there. But this will happen infrequently. So some of your chains will stop at this first one. Some of your chains will be able to go past this first one. And then the next time, maybe the next time the polymerase sees a T, it might grab one of these low concentration DTATPs here. And so you're going to have, this test tube will have a series of these fragments of different sizes, of all these different sizes. It will have these 4 different fragments that will be run out on the gel and separated. So you take each one of these tubes. So here would be your DDATP tube, your DDGTP tube, and GDCTP tube, and you will run these on the gel. And the gel that you're running is obviously sensitive enough to be able to separate those fragments that are only different even in one base different between each other. And so traditionally, this resulting fragments of each of these separate test tubes were run on a gel in different lanes like this, but so that the different fragment sizes could be spread out and the gel could be read. So let me actually at this point share one of the videos with you guys. And then just to help you further, there's also now a more modern way of doing it, and we can watch that one together too.
So what are the basics of Sanger sequencing? Let's take a look at our lab book. It all starts by having a short primer binding next to the region of interest. In the presence of the 4 nucleotides, the polymerase will extend the primer by adding on the complementary nucleotide from the template DNA strand. To find the exact composition of the DNA sequence, we need to bring this reaction to a defined stop that allows us to identify the base at the very end of this particular DNA fragment. Sanger did this by removing an oxygen atom from the ribonucleotide. Such a nucleotide is called a dideoxide nucleotide. This is analogous of throwing a wrench into a gear. The polymerase enzyme can no longer add normal nucleotides into this DNA chain. The extension is stopped, and we now need to identify what it is. We identify the chain terminating nucleotide by a specific fluorescent dye, 4 specific colors to be exact. Sanger sequencing results in the formation of extension products of various lengths terminated with dideoxide nucleotides at the 3' end. The extension products are then separated by capillary electrophoresis, or CE. The molecules are injected by an electrical current into a long glass capillary filled with a gel polymer. And during CE, an electrical field is applied so that the negatively charged DNA fragments move toward the positive electrode. The speed at which the DNA fragment migrates through the medium is inversely proportional to its molecular weight. This process can separate the extension products by size at a resolution of 1 base. A laser then excites the dye-labeled DNA fragments as they pass through a tiny window at the end of the capillary. And the excited dye emits a light at a characteristic wavelength that is detected by a light sensor. Software can then interpret the detected signal and translate it into a base call. When the sequencing reaction is performed, in the presence of all 4 terminated nucleotides, you eventually get a pool of DNA fragments that are measured and separated base by base. What you will get in the end is a data file showing the sequence of the DNA in a colorful electropherogram, and a text file which you can use to answer the questions you may be asking. And that, in a nutshell, is Sanger sequencing. For more information, download the free DNA sequencing guide through the link below.
All right, so in the second video, what it was showing that instead of you can actually run all of these if you have them instead of being labeled with a radioactive component. If you have it labeled with a different colored fluorescent component, then what happens is that you can actually run the whole mixture, like all 4 tubes can then be mixed and ran at the same time on one single lane, or in that case, in a capillary electrophoresis, so a very thin tube filled with agarose gel. And then as the current is applied, these bands will separate, and then a laser beam can actually detect what wavelength of light is being emitted by the different colored fluorescent probes that are on each one of these fragments, and as a result give you a sequence output. So it's actually a very clever way of figuring out the sequence of the DNA. So especially for DNA sequencing, I think this idea takes a while for you to sort of grasp and digest. So I really encourage everybody to go ahead and read through my notes again carefully at your own pace, really try and understand and figure out why it is that you're getting these 4 different fragments and how they correspond to the sequence that you can then read off of here, and how this sequence then relates to that sequence there. So yeah, do this on your own again, please, I've gone through it, and watch the videos again if I think they will help you. And make sure you understand just exactly how DNA sequencing makes use of these dideoxy analogs with their missing hydroxyl group, and how that is used in order to figure out the sequence of your DNA fragment. So I think that brings us to the end of our lesson here. Our next lesson, lesson 12, will be talking about a very important and very interesting topic of the social, ethical, and legal implications of biotechnology. So if nobody has any questions, I'm okay to end our lesson here at this point. I think I saw a question being asked. Somebody was asking a question earlier on in the chat about will you be receiving a review worksheet for your midterm exam? And the answer to that is that your unit assignments that you have had to do and hand in are the best form of review. And in addition to that, I hope that you have been trying out and working on these homework questions. And again, these are both these questions and your assignment questions go a long way to preparing you for your midterm exam. So I think what I will do is during our tutorial review session, I will be happy to go over any of the sections that we can review entire sections. If somebody doesn't understand something, say somebody comes and says, Miss, can you please explain gel electrophoresis to us again? Yes, I can do that. But we can also take up specific homework questions. For example, you might come and say, can we go over this homework question? And we can certainly do that. Or can we go over one of the assignment questions? Now, whenever I mark assignments and I give it back to you, I make sure to write notes on there for you so that you can figure out where you went wrong. But if sometimes my notes or the way I graded it isn't enough for you to understand how you can improve on that answer, how you can answer that question more correctly, please don't hesitate. Please bring it up, and we can go through it. We can go through it certainly during our review session, our tutorial session, which will be held before we have our exam, but also anytime really, if you like, just reach out to me and we can go through these things either before a lesson, or we can stay after a lesson and talk about something. But so yeah, we're one more lesson away. Next Monday, lesson 12, and then next Wednesday we can have our tutorial session. And then the following Monday after that, the 31st, we will be holding our midterm exam. Okay, everybody? All right, so if there's no other questions, I think we can conclude today's lesson.
Thank you. Good night.
Okay, thank you very much. Have a good evening.
Bye, Miss. Have a good night.